Today we'll be talking about biomolecules or macromolecules. This will be found in chapter 3 of your textbook. Here's an excerpt from the study guide. So carbon. Organic compounds contain both carbon and hydrogen, yes. So in order for a molecule to be classified as organic, it must contain both hydrogen and carbon. Inorganic molecules do not have both of these things. So this is kind of review. So by sharing electrons, carbon can bind to 4 other atoms. We talked about that in the chemistry section. And by doing so, it can branch in up to 4 directions. So remember, there were 4 electrons missing from its valence shell, therefore allowing it to make bonds with 4 other atoms. And so what we see here is methane, so it's just a carbon bound to 4 hydrogens. And so it makes a very symmetrical model or symmetrical shape, therefore making it nonpolar. Yeah, we call it a tetrahedron. Okay, so macromolecules. The 4 different macromolecules we're going to talk about today are carbs, carbohydrates, lipids, proteins, and nucleic acids. And so in the parentheses here, you can kind of use sugars are a type of carbohydrate. Lipids are composed of glycerol and fatty acids. Proteins are composed of amino acids. And nucleic acids are composed of nucleotides. And so I guess we usually do classify sugars as a type of carbohydrate, but when we're talking about the macromolecule of carbohydrates, usually it's a chain of sugars. So these are all of the polymers, or so polymers. They're formed from many of the same building block. So these are the polymers. So carbs, polymer, lipids, polymer, proteins, polymer, nucleic acids, polymer, and then the monomers are sugars, for example, amino acids, nucleotides. Lipids are a little bit special because you have lipids that your lipids don't always contain the same subunits. Okay, so the building blocks of polymers are called monomers. So poly meaning many and mono meaning one. All right, so a type of reaction that we'll probably talk about a lot through the quarter is dehydration synthesis and hydrolysis. So dehydration synthesis, that's going to be the process that forms a thing. It's going to synthesize something. It's going to take smaller parts and splice them together to make a larger thing. Whereas hydrolysis, this is where you're going to break things apart. So to break down hydrolysis, you can break it into hydro and lysis. So lysis means to cut, and then hydro, you could kind of associate hydro with water, so you're adding water to cut something. Dehydration synthesis, you can break this down into removing water and then making something larger, or making a larger thing from smaller things. So dehydration synthesis removes water, makes bonds. So here's an example of it. You have, here's a monomer, here's a monomer, here's a monomer, and it looks like you have 2 linked together, and then you have 1 solo monomer. And what happens is you remove this water, so hydrogen from here and a hydroxide group from here, and in the end what you're doing is you're splicing them together to make a larger molecule of 3 subunits. Now hydrolysis, you're adding water and you're breaking bonds. So here you've got this 3 subunit chain of whatever, and you're adding water and it breaks apart that larger thing into 2 smaller things. So most biological reactions of this sort are mediated by enzymes. So usually what's going to catalyze these reactions are enzymes. And for the most part, enzymes are, one, they're biological catalysts. So, and to define the term catalyst, a catalyst is just something that lowers the activation energy or promotes a type of reaction. Enzymes are biological catalysts, so those are usually going to be found in living things, and for the most part, they're composed of proteins. So most enzymes are composed of proteins. So enzymes, they'll speed up chemical reactions in cells. All right, so carbohydrates. So you've got simple carbohydrates or single sugar carbohydrates. You have disaccharides. So, or you could actually say monosaccharides. So a single sugar would be a monosaccharide. Two sugars bound together. It's a disaccharide. So di meaning two. And then polysaccharide, those are three or more sugars. And usually all of your sugars are going to end in the word ose. So glucose, fructose, you splice them together, you got sucrose. So simple sugar monomers, monosaccharides, glucose and fructose. So the common formula for all sugars, for all sugars for this specific PowerPoint lecture is C6H12O6. When you're talking sugars, if you see this ratio, C6H12O6, you're typically going to be talking about some type of sugar. So glucose is a monomer, fructose is a monomer, and sucrose, that's the joint of the two. So this is your disaccharide. And notice they all end in ose. Glucose, fructose, sucrose, great. This is just going to show you the process of probably dehydration, synthesis, and hydrolysis, if I remember correctly. Okay, so more on carbohydrates. So disaccharides, two things. Sucrose, remember that's glucose, and fructose put together. Maltose is going to be glucose plus glucose. Polysaccharides. Oh, another thing I forgot to mention is glucose and fructose, they're both sugars. They both have the same chemical formula. However, if you look at their chemical structure, the atoms are actually arranged differently. So if you look at this ring, 1, 2, 3, 4, you got 4 carbons in this ring, and you have 1, 2, 3, 4, 5 carbons in this ring. The other carbon's up here. No. Well, here's one of the carbons, and then here's the other carbon. So you have the same chemical formula, different arrangement of atoms. We call those isomers. So these are isomers of each other. Okay, so your disaccharides, you've got your glucose and fructose, and your maltose is where you have glucose bound to glucose. Polysaccharides, those are going to be your complex carbohydrates. And so starch, that's the way your plants will store sugar. Glycogen, that's the way animals will store their sugar. So we usually store it in our liver and our muscles. And then cellulose, this is actually indigestible sugars. So the bonds that link each monomer sugar together is a little bit different than starch or glycogen, for example. And we don't contain the enzyme, or we don't produce the enzyme ourselves to digest cellulose. And so we usually consider cellulose to be that insoluble or soluble fiber. So this is our source of fiber or roughage. Cellulose is actually used most commonly as plant cell walls. So that's what plants will, and we'll get into that, your plant cell walls in the next chapter. And then we also have chitin. This is a polysaccharide used by insects and crustaceans to build their exoskeleton. Also forms the cell wall in fungi. So the big difference to note here that usually confuses people is the difference between starch and glycogen. So starch, that's what's going to be the plant's way of storing their sugars. So that's the form that plants will store their sugars, whereas glycogen, that's the way animals will store their sugars. Okay, so the storage form of carbohydrates is what in animals and what in plants. So in animals, glycogen. In plants, starch. All right, on to lipids. A little bit of a special macromolecule, just because they don't have a consistent single subunit. So usually lipids are going to be nonpolar. So if you look at this structure here, remember how methane was nonpolar because it was symmetrical. And what you essentially had was a carbon in the center and 4 hydrogens on the outside, and there was no polarity to the molecule, and all of the electrons, even though the electrons were shared in an uneven way between hydrogen and carbon, it was symmetrical, therefore making it nonpolar. Well, in this case, you have more symmetry than asymmetry, and that's what makes this nonpolar. And the disparity in the electronegativity between your hydrogen and carbon is so low that it's still less polar. Okay, so nonpolar stuff. Also, another thing that I forgot to mention is, or that I didn't forget to mention, but I want to remind you, is that polar things have a tendency to dissolve other polar things. Nonpolar things have the tendency to dissolve other nonpolar things. So the term that is commonly used is like dissolves like. That's why when you take, your oil and you mix it in with water, they separate. You've got water, which is polar, and you've got your fat or your oil, which is nonpolar. And so usually they're repulsed by each other. They don't dissolve each other. And so you don't get this homogeneous mixture when you mix them together. Okay. So back to lipids, or back to lipids, yeah. Triglycerides. So you've got oils and fats. Oils are typically going to be liquid at room temperature, and your fats are going to typically be solid at room temperature. Typically your oils, when you consider something to be an oil, it's composed of many unsaturated fats, many unsaturated fats, and your fats, or your animal fat, you could say animal fat, is typically going to be composed of many saturated fats. And then we also have our phospholipids of membranes. So those are going to be the things that we use to compose our cell membranes. And then we also have cholesterol, which is another special case. So like I said, lipids are kind of a special case. This is more of like a gray area when it comes to defining things. So let's look at triglycerides first. So the basic structure of a triglyceride is, and so this is your dietary fat. When you're consuming fats, typically they're going to be in the form of triglycerides. So tri meaning 3, glyceride meaning glycerol or glyceride. So you're going to have one glycerol molecule, and that's going to be at the top here. This is your glycerol, so this is kind of like the backbone of your triglyceride. And then you have 3 fatty acid hydrocarbon chains. So hydrocarbon, these are hydrogens, and these are carbons, hydrocarbon chains. And the chains of carbons, they are chains of carbons with hydrogens sticking out. So this is a typical triglyceride. You've got your glycerol backbone, and then you have 3 fatty acid chains hanging off of it. Okay, so the difference between saturated fats and unsaturated fats when you're looking at the chemical structure is your saturated fats are going to be fully saturated with hydrogens. So if you look here at this hydrocarbon chain, you've got carbon, 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 and so on, and every single one of these carbons are going to be fully saturated with hydrogens. Now, we know that from chemistry that if you create a double bond here, for example, if you create a double bond here, that means that you have to remove bonds from something else to establish that double bond. So if you were to create this double bond here, you would have to remove one of these hydrogens from here, And so if you're unsaturating it with carbons, or with hydrogens. So if you're desaturating it essentially of its hydrogens to form that double bond, now we call it an unsaturated fat. So in this case, you have one double bond which yields this little kink here. So we call this a mono unsaturated fat. One double bond makes it a mono unsaturated fat. So mono unsaturated fat has only one double bond. So things like canola oil, peanut oil, olive oil, those are going to have a lot of mono unsaturated fats. And polyunsaturated fats have many double bonds. So if you have two or more double bonds, now you're a polyunsaturated fat. And so the way we determine the omega numbers, so omega-3 for example, you just go to the tail of the fat and you count how many carbons up until the first double bond. So 1, 2, 3. This is an omega-3 fatty acid. Okay, so trans fats, those are that is an unsaturated fat that has an arrangement of carbons that makes it behave in the body more like saturated fat. So let's see if we have a trans fat. So as you'll probably learn in chemistry, there's a difference between cis bonds and trans bonds. This is going to be more pertinent when you get into organic chemistry. So to break this down simply for you guys, cis means same, trans means different. And so when you have this cis double bond, if you notice here, from this carbon to the left, everything's at a downward angle. And from this carbon to the right, everything's at a downward angle. And so that means, so cis meaning same, you've got the same direction from this double bond. Now when you're talking about trans, so from this carbon to the left, it just goes straight. And from this carbon to the right, it goes, I guess you could, if you tilted it up this way, this carbon would go down and this carbon from this carbon to the right would go up. And so you'd have, you know, a mismatch in the angle from that carbon to the left and from that carbon to the right. What makes trans fats bad is because our body treats them as though they are, well, our body, they behave a lot like saturated fats. However, they are unsaturated fats. And typically we'll integrate this stuff into our cell membranes. And because they act as saturated fats, remember saturated fats are typically more solid at room temperature. They have a little bit of a hardness to them. They'll actually make the cell membranes of our cells a little bit more stiff compared to just a regular unsaturated fat. Okay. Yeah, so this is just saying, you know, it's bad to eat trans fats and saturated fats. I usually have a slide between here, but oh well. Okay. Now lipids, or back to, not lipids, phospholipids. So phospholipids, those are going to be the main component of your cell membrane. And so you're going to see something common here. You've got that glycerol backbone. You've got a fatty acid tail or hydrocarbon chain fatty acid tail here. You got two, but instead of the third one, which would make it a triglyceride, in place of that you have this choline head, which is polar. And so if you look at this phospholipid bilayer here, the polar, so remember choline is polar, the polar heads sit at the outside and the non-polar tails sit on the inside. And so if you think about the environment of our cells, typically they're drenched in water. They're surrounded by water. And so remember, like dissolves like. Your polar stuff wants to be around other polar stuff. Your non-polar stuff wants to be around other non-polar stuff. Well, that's why all of your phospholipid tails will actually congregate in the center near other non-polar things. And then your phospholipid heads will congregate towards the outside, towards water. So polar is attracted to polar, non-polar is attracted to non-polar. Some other characteristics of your phospholipid bilayer, which we'll probably talk about in more detail later. You've got things like carbohydrate chains. So these could be like tags for your cells to tell other friendly cells, hey, I'm friendly. You have things like cholesterol, which we'll get into a little bit later. And then you also have membrane proteins. So this is the way you get things in and out of your cells. This is how your cell is sensing the outside environment of the cell. Or it could contribute to the cell's ability to sense the outside world. Okay, now lastly we have, I think it's lastly, we have cholesterol. Yeah, it is lastly. So your cholesterol, in short, are your fat carriers. So they're going to be involved in transporting fats from one area of your body to another area of your body. And what I want you to remember here is that HDL, so high density lipoproteins, those are your healthy cholesterol. That is your healthy cholesterol. And your LDLs, low density lipoproteins, that is actually your bad cholesterol. You don't want high LDL. You want high HDL and low LDL. And so if we look at so cholesterol is a type of lipid, but look at the structure. Way different from the other stuff. That's why I say this is kind of a special case. Okay. If you were to add olive oil to your food as a part of a diet to lower your risk of atherosclerotic disease, you would use olive oil that is what? So atherosclerotic disease, that is the hardening of your blood vessels. Typically your saturated fats and cholesterol will contribute to this, bad cholesterol. And so if you want to reduce the risk of that, you're going to want to have higher intake of unsaturated fat. So you're going to use olive oil that is hydrogenated. So if you were to hydrogenate a fat, you're essentially you're adding hydrogens to it, so you're saturating it artificially. If it's liquid at room temperature, likely it is composed mostly of polyunsaturated fats. So this is the correct answer. But let's look at the other ones. Is modified to be solid at room temperature. That's pretty much where we've made saturated fats, and has lard added to it. That's not good for you. This lard contains a lot of saturated fat. So B would be the correct answer. Okay, proteins. So protein is a polymer built from various combinations of 20 amino acid monomers. So amino acids are the monomer, a protein is the polymer. The functions of proteins that you create within your cells are determined by their three-dimensional structure. So they're unique structures. Their 3D structure is what gives them their function. Enzymes are an example of proteins that serve as metabolic catalysts. We talked about this earlier. And what catalysts do, or the metabolic catalysts do, is they regulate the chemical reactions within cells. So your basic skeletal structure of an amino acid. So we're going to go to amino acids, the monomer of proteins. So amino acids are the building blocks of proteins. They have an amino group, a carboxyl group, and an R group. So our amino group, this is our amino group. Here's our carboxyl group. And then our R group is kind of a placeholder for the various R groups of your amino acids. So remember, we have 20 different types of amino acids. What's consistent between all of them is their amino group, their carboxyl group, this alpha carbon. What makes them different, what differentiates them from each other, is their R group. So here we've got an amino acid, here we've got an amino acid. We splice them together to make a polypeptide, poly meaning multiple, peptide meaning having to do with proteins, so polypeptide chain. And so this type of reaction, we're removing water. This is dehydration synthesis. Okay, so both of these are covalently bonded to a central carbon atom. So here's your alpha carbon, or your central carbon atom. Also bound to this central carbon is a hydrogen atom, great. And varying depending on amino acid, the chemical group symbolized as R. So R, like I said, is a placeholder for your different letters of the amino acid alphabet. Let's take a look at these 3 amino acids, leucine, serine, and aspartic acid. Right off the bat, you should notice that their skeletal structures are actually all the same. So they all have that amine group, the carboxylic acid, the alpha carbon bound to the hydrogen. And what differentiates them from each other are their R groups. So those are going to be resembled or highlighted by this peach color here. And one thing that will likely be helpful for you in the future is making the distinction between polar and non-polar R groups, or polar versus non-polar amino acids. So if you're looking at these R groups, this one kind of closely resembles the fatty acid tails that we saw when we were looking over lipids. And so this one would actually be non-polar. Now these two, they actually have, as a general rule of thumb, if you see a charge anywhere on a molecule, likely it's going to be polar. And so here you have this oxygen with a negative charge. So for one, it's got an extra electron, but it's also going to be pulling in. So oxygen's far more electronegative than carbon, and so the electrons are actually going to be pulled towards this oxygen more than it is towards that carbon. And likewise for this oxygen as well. And so that's going to make this R group polar. And the same thing kind of goes for this R group as well on serine. So you have this carbon bound to this oxygen, and this oxygen, because it's more electronegative, is going to be pulling those electrons closer towards it. Now, we talked about non-polar and polar, and we've also talked about how like dissolves like, and how polar things like to be around polar things and non-polar things do not like to be around polar things. We've also talked about how water is polar. So, taking into consideration the polarity of these amino acids, we can infer whether or not they're going to like being around water or not. So we know that when we look at the term hydrophobic, phobic means afraid of, and philic means, so if we're looking at hydrophilic, philic means lover, a lover of, or loving of that thing. And so hydro, we can associate that with water. So water fearing, or hydrophobic, and water loving, or hydrophilic. So these two are polar, therefore hydrophilic. This is nonpolar, therefore hydrophobic. It's not going to want to be around water because it is nonpolar, and water is polar. These want to be around water because water is polar, and these are also polar. Okay, so amino acid monomers, they're linked together to form polypeptides, so many peptides. This is accomplished by a dehydration synthesis reaction, mediated likely by an enzyme. So this links the carboxyl group of one amino acid to the amino group of the next amino acid. So that's backbone attaching to another backbone. So you've got your carboxyl group, remember you have this hydroxyl within that, and then within this amino group, you've got this hydrogen. You remove this hydroxyl group and this hydrogen, you combine them together, you're forming water, and you're dehydrating the molecules. You're taking away water from the molecules to form a bond between them. So you're splicing together the backbones of these various amino acids. Okay, so protein structures. So your primary structure, when we're talking about primary structure of amino acids, this is going to be the linear sequence of amino acids. So in this case, you're making a dipeptide here. You're linking one amino acid to another amino acid. Here's your first amino acid, here's your second amino acid, and so you've got amino acid 1, amino acid 2, and let's just say we had more amino acids, amino acid 3, 4, 5. That would be the linear sequence of amino acids. That would be your primary structure. Now, when we're talking about our secondary structure, we're talking about how the backbone will actually interact with itself. And so what happens when you get these long strings of amino acids to form these long proteins? Typically, what's going to happen, and if we can, I can't zoom in because I don't want to screw up the presentation view. But typically what's going to happen is the oxygen in the backbone is going to form hydrogen bonds with the hydrogen within the backbone. And so in this case specifically, you're forming, if you get a series of these forming, you're creating something called a beta-pleated sheet. And so this is just where the backbone folds in on itself and interacts with itself. Another way your backbone can interact with itself is through forming an alpha helix as well. So you can get a beta-pleated sheet, you can get an alpha helix. Those are two different formations that are described by secondary structure. And so you're still getting hydrogen bonds. So backbone on backbone action, that's your secondary structure. Now, as you might presume, we're going to, so this is actually, this is a closer view of it. So we actually future-proofed myself, I guess. So yeah, this oxygen is very attracted to this hydrogen attracted to this oxygen, and your backbone is attracted to the backbone. Your R groups just dangling off. But as you might suspect, there are more structures. So there's primary structure, which is the linear sequence, secondary structure, which involves the interaction of the backbone and itself, and then you have tertiary structure. So this is where your R groups will actually interact with each other. And so remember, our R groups are going to have various characteristics. Those characteristics are determined by their chemical structure, and that chemical structure determines whether or not it's polar or nonpolar, and therefore hydrophobic or hydrophilic, or hydrophobic, respectively. So tertiary structure is R groups. In sheets, helices, they will interact with each other. So they can form hydrogen bonds between one another. Let's see if I created, no, that's not a good representation. Okay, well, if you've got R groups interacting with each other, we call that tertiary structure. And if you have your, or actually, so once we complete our tertiary structure, we get our primary structure, which is the linear sequence of amino acids. You get the backbone interacting with itself, and then you also have the R groups interacting with other R groups. You form a blob, a protein blob. Now that protein blob can interact with other protein blobs, and so if you get tertiary subunits interacting with other tertiary subunits, with other protein blobs, now we're talking about quaternary structure. Okay, this is what I was trying to get to. So what kind of determines your protein folding, the three-dimensional shape of your protein, is kind of your R groups. So all of your hydrophilic R groups are going to want to hang out towards the outside of the protein. But now if you look on the inside of this protein, here's all of your hydrophobic R groups. And so there's this constant battle of reaching equilibrium where you're stable with your hydrophilic R groups hanging out on the outside, and our hydrophobic R groups scrunched up on the inside with other hydrophobic R groups. So we got tons of different types of proteins. We've got structural proteins, so proteins that would contribute to, let's say, the structure of your cell, so like your cytoskeleton. You have storage proteins, so hemoglobin is a good example that transports, your hemoglobin will transport oxygen. We also have contractile proteins, so your muscles contract, and that involves the sliding of your cytoskeleton across one another, and that's determined by, or that is conducted by proteins. We have defensive proteins, so things like antibodies, transport proteins, so on the cell surface you might need to transport ions into your cell or move stuff out of the cell. And you also have signaling proteins, so things like neurotransmitters, the way our neurons communicate with each other, those could be made of proteins. Enzymes, so the catalysts within the cell, the things that move all of your metabolic processes forward, composed of proteins. So proteins are kind of vital for life essentially. All right, so question. Proteins differ from one another because A, the sequence of amino acids in the polypeptide chain differs from protein to protein. Each protein contains its own unique sequence of sugar molecules. The number of nucleotides found in each protein varies from molecule to molecule. And the peptide bonds linking amino acids differ from protein to protein. Okay. So the best answer here, actually the only answer here, is A. The sequence of amino acids in the polypeptide chain differs from protein to protein. What makes a protein differ from another protein is the sequence of amino acids. What makes an amino acid different from another amino acid is the R group. So the peptide bonds are all going to be the same. Proteins, so number of nucleotides, we haven't even gotten to nucleotides yet. So nucleotides, that's a different monomer for a different macromolecule. And then sugars, those are going to be involved in composing, or those are going to compose your carbohydrates. Okay, so our last macromolecule we're going to talk about is nucleic acids. So DNA is the prime example of a nucleic acid, and it's composed of monomers called nucleotides. So the monomers for nucleic acids, such as DNA, are nucleotides. So nucleotides have three parts. So kind of like our amino acids, they've got a skeletal structure that is consistent among all nucleotides. So 5-carbon sugar called ribose in RNA and deoxyribose in DNA. So later on in the quarter, we're going to talk about the difference between RNA and DNA. RNA is ribonucleic acid, and then DNA is deoxyribonucleic acid. We don't really have to get into the details of that right now, though. Okay, so they have a 5-carbon sugar. So here's your 5-carbon sugar. And you've got a phosphate group. So here's your phosphate group. So P for phosphate. And then you have a nitrogenous base. So this is going to be kind of akin to the R groups in your amino acids. This is going to be the letter of the nucleotide alphabet. But with nucleotides, there's only 4 letters compared to amino acids, where there's 20 different letters. A nucleic acid polymer, a polynucleotide, forms from the nucleotide monomer, from the nucleotide monomers when the phosphate of one nucleotide bonds to the sugar of the next nucleotide. So what this is saying is that the sugar of one will bond with the phosphate of the other, of another one, forming the chain. So the result is a repeating sugar-phosphate backbone with protruding nitrogenous bases. And so here you have your sugar, which is bound to the phosphate, which is bound to the sugar, which is bound to the phosphate, and so on. And then on the inside, you have dangling your nitrogenous bases. So you've got, adenine as an example, thymine, cytosine, guanine. And so those are going to be your 4 different letters for your your nucleotides. Another thing that we need to know is that adenine is going to bind to thymine, and guanine is going to bind to cytosine. So there's a few ways to remember this. You got apples and trees. So, trees and apples. And then you have guanine and cytosine. So, might be outdated, but girls and cats. Or you could just even look at the first letter. Guanine kind of looks like, G kind of looks like C. So it looks like we will be needing to know the difference between RNA and DNA. At this point, we'll probably touch on it more later. So RNA, the difference for now, the main difference that I want you to know between DNA and RNA is that DNA is going to contain adenine, thymine, guanine, and cytosine. So those are the 4 letters for your DNA. Now when we're talking about RNA, you've got guanine, which still binds to cytosine, but you have adenine, and in place of thymine, you have uracil. Typically, RNA is going to be found in single-stranded units, at least when we're looking at, our cells, for example. And DNA is going to be found paired with another strand of DNA, so double-stranded DNA, whereas RNA will typically be single-stranded within our cells. Another thing we're going to look at is ATP. So ATP, or adenosine triphosphate, this is our energy currency for the cell. All organisms require ATP to drive forward all of their cellular processes, and without ATP, things just die. And so if you look at it here, it looks a lot like the subcomponent of DNA. So you've got a 5-carbon sugar ribose, or actually this would be RNA. So you've got your 5-carbon sugar ribose bound to your nitrogenous base, adenine. So adenine triphosphate. So instead of one phosphate, which can bind, if we're dealing with RNA, bind to another 5-carbon sugar, you've got 2 other phosphate groups tagged onto it. So you have adenosine triphosphate. Okay, so question. Which of the following statements regarding nucleotides is false? Nucleotides contain nitrogenous bases. Nucleotides contain sugar molecules. Nucleotides contain lipids. Nucleotides can be linked together to form nucleic acids. So A is correct. They contain nitrogenous bases. That's your R group, essentially. Nucleotides contain sugar molecules. Yeah, your deoxyribose or your ribose sugar, 5-carbon sugar. They do not contain lipids, so C would be the correct answer. And nucleotides can be linked together to form nucleic acids. Oh, got another question. So glucose molecules are to starch as blank are to proteins. Glucose molecules are to starch as blank are to proteins. So glucose is the monomer of starch. What's the monomer to proteins? That's amino acids, so C would be the correct answer here. All right, that's it for today. Took